Howdy! In this video, we are going to go over some of the different pheromones of the honeybee, where they're produced, and their uses and functions. Well, what is a pheromone? A pheromone is a type of semiochemical, or chemical used for communication. There are two different types of semiochemicals: allelochemicals, which are used for interspecific communication, or communication between species, and pheromones, which are used for intraspecific communication, which is communication within species. Pheromones are produced in the secretory glands. The secretory glands found on the head of the honeybee are the mandibular glands, hypopharyngeal glands, and the salivary gland. The thorax holds the salivary glands and the Arnhart glands, while the abdomen is home to Dufour's gland, Nasonov gland, and the sting gland.
A pheromone's ability to travel is dependent upon its volatility, which is basically the chemical's solubility in air. Compounds with a high volatility can travel further more quickly and can be detected and followed to their source from a greater distance. They are usually less specific compounds and have a low molecular weight. Compounds with low volatility can only travel short distances and are usually spread by only contact. Generally, these are more specific compounds and have a higher molecular weight, which prevent them from traveling very far. Pheromones act as either a primer or a releaser. Primers take time to cause an effect. They may result in a change of hormone production over time, lead to behavior change over time, or have a long-term effect gradient. Releasers, on the other hand, cause immediate change of behavior. Dufour's gland produces the egg-marking pheromone, which is then secreted into the vaginal cavity upon egg laying. This allows for distinction between eggs laid by the queen and eggs laid by her workers. There's also a difference in the queen egg-marking pheromone and the worker egg-marking pheromone in queen-right colonies and queenless colonies. Queen-right colonies contain long-chained alkanes, and queenless colonies contain long-chained esters.
Produced in the Arnhart glands, the trail pheromone is also produced at the tip of the abdomen, which trails the ground as the bee walks. It has an attractive effect on other bees, and it marks the flowers that have already been visited by the bee. It can also work in concert with QMP to inhibit queen rearing by letting the workers know not to build queen cells in the areas she has already visited. Brood pheromone is a blend of 10 fatty acid esters, some of which are produced in the salivary glands. It is present in different proportions based on age and caste of the larvae. This pheromone regulates pollen foraging by the colony and stimulates workers' hypopharyngeal glands, which then in turn stimulates food production. It has low volatility and is transferred through contact. It can work in concert with E-beta ocimine to inhibit ovary development in the workers. Basically serving as a homing scent, the scent pheromone attracts bees to the location where the scent is being produced. It is produced in the bee's Nasonov gland, which is only present in worker bees, so queen bees and drones do not produce this pheromone. Workers will expose their Nasonov gland and begin fanning in many instances, such as at foraging sites, at the hive entrance, at a swarm cluster, at a new nest site, and it can work in concert with the 9-ODA component of QMP to facilitate clustering around the queen during swarming.
The queen mandibular pheromone is a blend of 5 components produced in the mandibular glands of the queen. It has low volatility and is transferred mainly by contact. It has a releaser effect on retinue behavior, causing worker bees aged 7 to 11 days in the vicinity of the queen to respond and tend to the queen by grooming her. Messenger bees also exchange QMP with other workers in the colony. Effect by inhibiting queen rearing in the colony. When the levels of QMP drop, the colony begins to rear queens. When QMP levels are high, it can also suppress the ovarial development of the workers.
The alarm pheromone, 2-heptanone, is produced in the mandibular glands of older bees. These bees will assume an aggressive posture and charge towards the source of the alarm when releasing this pheromone. It is a releaser pheromone that causes instant reaction by the other bees nearer. The release of this alarm pheromone is a pre-stinging behavior. Many other arthropods and social insects use similar chemicals as primitive alarm systems. The sting pheromone is made up of more than 80 chemicals that come from the sting gland and the Koshchev gland. The most important chemical is isopentyl acetate. The most abundant chemical is Z11-eicosanols. The release of this pheromone attracts bees to sting all in one area. Combining Z11-eicosanols and isopentyl acetate leads to a synergistic response. The forager pheromones are not really identified. However, removing foragers from the colony decreases the age of foraging onset in the young bees because they have to replace the removed foragers. Removing young bees delays the onset of foraging in the young because there aren't that many young to feed anymore. If you wash foragers in hexane and remove all of that extract and then put it on a new colony, it will increase the age of foraging because they think there are more foragers present. These are our references. Thanks, and gig 'em.